Okay, so now we're going to talk about how genetics impacts cancer. Um, we'll start with epigenetics. If you remember that movie about the ghost in our genes, you know that methyl tags play a very, very important role in whether genes are allowed to express themselves or not and code for their proteins. Um, oncogenes are the ones that accelerate cell division. If we take methyl tags off of these oncogenes, they will suddenly start to express themselves inappropriately. They will overexpress themselves. Global hypomethylation is a phenomenon that occurs with aging. As a person ages, different methyl tags on their genes will wear off, and that can contribute to risk. That's why you see the development of cancer in older people generally. Although there are childhood cancers, definitely. Um, they can also be induced by environmental exposure, such as exposure to alcohol use, repeated alcohol abuse in head and neck cancers. The tissues in those regions, the cells will lose the methyl tags on their oncogenes. Histone modification is another way that we can alter the expression of genes, and generally, that means that the histone protein is wound tighter and it silences those suppressor genes and the DNA repair genes. They're present. Um, the original genome had mechanisms, but as a person is exposed to different environmental agents, sometimes those histones will be modified. You see this particularly in inflammation. Um, microRNA downregulation also can cause overexpression of oncogenes. Um, and this is good to know because cancer treatment can look at the epigenome and use agents that selectively methylate the oncogenes. That's sort of in development. So research is underway to investigate the effects of histone modifying agents that can silence oncogenes and how antioxidants might work um, to promote methylation of oncogenes as well, or demethylate your suppressor genes. Um, So that's just a little bit about epigenetics. Aging and cancer. So we talked about global hypomethylation of cells can encourage overexpression of oncogenes. Also, as people age, genomic instability contributes to cancer risk. The more times a cell has to divide, the greater the chance they're going to make a mistake. So by the person gets to be 90, their cells have reproduced many, many times. With each reproduction, the process is a little less precise. um, I mean, it was built to last, the genome, and that's why we have all those protective structures like telomeres. But the telomeres get shorter as people age. It's one of the markers of biological aging as opposed to chronological aging is telomere length. It's not the only one, but it is an important one. Cumulative environmental exposures. I mean, somebody who's been alive for 80 years has been exposed to air pollution for a good number of them, has been exposed to maybe more sunlight, more asbestos, more tobacco smoke, more carcinogenic substances, more viruses. In the elderly, we have decreased immune function. So if the immune system can't identify and kill those cancer cells, because it isn't working as effectively as it was, we can have increased risk for cancers. So telomeres are those little end caps on each chromosome. That happens somewhere in week 2. I think we talked about it. Everything circles back. It's all useful. Disrupted function leads to damaged DNA. So telomere length is influenced by both genetic factors. Some people seem to be programmed for longevity. When population studies were first done on genome-wide um, the genome-wide association studies. They studied um, Ashkenazi Jews because there is a lack of heterogeneity in that population. Um, and they found that certain mutations were associated with longevity, or certain variants were associated with longevity, and certain variants were associated with shorter lifespans. So some of that is influenced by genetic factors. Some of it is influenced by non-genetic factors, like smoking makes telomeres shorter, exercise makes them longer. Certain antioxidants might make them longer. Stress and repeated exposure to cortisol makes them shorter. Shorter telomeres are strongly linked with breast, bladder, esophageal, gastric, ovarian, and renal cancers. Um, Cancer cells, interestingly enough, because they're all about protecting themselves and avoiding apoptosis, they want to live forever, they're like vampires. They upregulate the production of an enzyme that promotes longer telomeres. So telomerase is that enzyme. Um, it's very interesting. So sporadic versus familial cancers go back to that idea of all cancers genetic, but only a little bit of it is hereditary. Most of the cancer you'll see will be sporadic cancer. It occurs in the absence of a strong family history. The rate of occurrence in the family of the proband is not higher than the general population. Um, this is due to somatic mutations in cells that alter the oncogene suppressor genes or DNA repair genes. They're not passed on to the children. And no genetic testing is available that will detect an increased risk on that person's genome. Genetic testing could be done on the cancer cells to determine characteristics that enable treatment. We can find out if they're receptive to estrogen um, and use medications like that. Um, but many of you in your pedigrees indicated that there were family histories of cancer. If it's all different types of cancer and the rate of cancer in your family is not higher than what you would expect, and people are diagnosed at later ages, it's more likely to be involved in sporadic cancer. In contrast, we have familial cancer or hereditary cancer. Now, you're not born with the cancer, you are born with a risk for cancer. But this occurs in families when you look at a pedigree. Now, if your pedigree had higher frequency, um, you look at it, and in every generation, 2 or 3 people have gotten the same types of cancers, melanoma, for example. Um, and it's the same type of melanoma, and it's higher than what you would expect in that general population. It's more likely to be familial. If you look at that pedigree and you see a specific pattern of inheritance, if you need help interpreting pedigrees, please go back to the recordings that I did on pedigree interpretation, where we look at different pedigrees and we see how diseases are passed on, and we can sort of interpret an inheritance pattern: autosomal dominant, autosomal recessive. Um, then your cancer is more likely to be familial. Um, if the cancer is diagnosed at younger ages than are typical of the general population. Now, breast cancer, generally speaking, most of the people who are diagnosed with it are women, and they are postmenopausal. Average age would be about 63 for diagnosis. But if you're seeing it diagnosed in people at the age of 34 or at 42, um, we're looking possibly at a familial cancer syndrome, especially if we're seeing it in genders that typically don't get that cancer. It is caused by germline mutation. It may have been caused many generations ago, but it is passed on. Genetic testing is available. Um, it could be complicated to make decisions based on that because of penetrance. Penetrance is the percentage of individuals with the gene variation who will actually get the disease. If it's a syndrome with high penetrance, like retinoblastoma, we know the risk is higher. If it's a syndrome with lower penetrance, it could be hard to counsel people about what their actual risk is. So let's kind of just reiterate what the difference is between sporadic and familial. And if I'm saying something more than once, you're very likely to see it again. So you might get a pedigree on the exam, and one individual has a cervical cancer. In this example, woman contracts HPV. She is our proband. Virus causes initiation of the cancer. Okay, so exposure to that virus is the initiating event. The abnormal cells are detected by pap smear when they're in that mild to moderate dysplasia stage. Provider performs a LEAP procedure, removes the cells. No other family members have this history of cervical cancer, and she's not going to pass it to her children. It is wise to vaccinate everyone against HPV because people are at risk. And if you can vaccinate them against cancer, why not do that? But would you necessarily indicate family members get tested for any particular gene variant? No. And do they need increased screening? not unless they have high-risk behaviors that expose them to HPV. So that's sporadic cancer. And if you were to see that in a pedigree and it was an isolated occurrence of that particular cancer, you wouldn't necessarily look for a genetic risk. Familial cancers are different. Now remember, they only account for up to 5 to 15% of known cancer diagnoses. As we know more about the genome, that number may rise, but at this moment in time, it's not the majority. So examples of familial cancer. A woman finds a lump in her left breast. She sees her provider. She's referred for testing, has her mammogram and an ultrasound, and then a biopsy. She's diagnosed with breast cancer. She's 36 years old. During this visit, she tells the provider that her mother and both maternal aunts have also had breast cancer. They were all diagnosed under the age of 50. Her maternal grandmother died of ovarian cancer, which was very advanced at the time of diagnosis, and she was also under the age of 50 when that happened. So if this woman came to a provider's office and said, I have this lump in my breast, and she's 36 years old, you might say, well, could be fibrocystic breast disease. But now she tells you this family history, and you're like, oh, okay. And that's when you maybe send her for testing. Honestly, anybody who has a lump in their breast should be evaluated, but much more likely to be cancerous if she's got that family history. Now she reports she's of Ashkenazi Jewish ancestry. Well, that increases her risk substantially, as it does for many other diseases. They test for BRCA1 and BRCA2 gene variants associated with cancer, and the woman ends up testing positive for the mutation on the BRCA1 gene. Her brother's also diagnosed with breast cancer. Atypical for men to be diagnosed with breast cancer, particularly in their 30s and 40s. However, with this BRCA mutation, men are more likely to develop breast cancer. The cancer is found to be triple negative, and that is common in BRCA1 variants. That it doesn't have estrogen receptors. There are 3 types of estrogen receptors. This cancer is triple negative. It has none of those. And that information guides treatment. So we're not going to give her an agent that blocks those estrogen receptors, those HER2 receptors, because it won't work. We're going to give her a different therapeutic agent. So this patient is capable of passing on the gene to her children. Because the gene variant has autosomal dominant inheritance, each child has a 50% chance of inheriting this defective allele, which, because it's dominant, has a pretty good chance of expressing itself. And that's going to guide our decision-making about testing family members. So let's talk about cancer syndrome. So when a gene change that has been identified greatly increases risk for the development of a particular cancer, it can be called a family cancer syndrome. And there are several of them. If you really want to, you can Google them. If you're developing your genetic disorders presentation and you want that information for your case study, go to it. But there are lots and lots of family cancer syndromes. We suspect a cancer syndrome when the following features are present. Many cases of the same type of cancers are in a pedigree, much more than you would expect for that type of cancer in the general population. So if you're looking at a pedigree and you've got 3 or 4 generations of good data, and we've got lots of individuals affected by a triple negative breast cancer, we know we have to look for family syndromes. If the cancer occurs at younger ages than usual, if more than one type of cancer is diagnosed in a single person, the person gets breast cancer at 42 and then develops ovarian cancer after a mastectomy, that is a pretty good tip-off. If cancers occur on both sides, so it's rare for, if we're having carcinoma in situ, for it to be in both breasts, if it's not a familial cancer. It's more common in cancer syndromes, both kidneys, both eyes. If you have 2 of something and they both have cancer, or you get cancer in one breast today and 5 years from now, after that treatment was successful, you now have contralateral breast cancer. That would indicate maybe a familial cancer syndrome. Cancer occurs in a sex not typically affected, for example, male breast cancer. Cancer occurs in many generations. The more generations you have data for, the better your decision making will be, the more information you have. More than one childhood cancer in siblings. Childhood cancer is rare to begin with. If you see the same cancer occur in a sibling in childhood, or you have more than one of them, you're looking at a familial cancer syndrome. So, some examples of cancer syndrome, and these could make great disorders for your genetic disorder presentation. So, if you're interested in any of them, let me know. Hereditary breast and ovarian cancer has an increased risk for breast and ovarian cancer. Some variants of BRCA2 are linked to familial melanoma. So if you're seeing a lot of breast and ovarian cancer and you're also seeing familial melanoma, or you're seeing a lot of melanoma, it's the same type. These are caused by alterations in BRCA1 and BRCA2 genes. BRCA genes are DNA repair genes. So there's an initiation, and the body's all set to like scan the genes. What's wrong with you? Did you pass our quality control? If we don't have good quality control on these genes, they can be allowed to proliferate. It is inherited in an autosomal dominant pattern, meaning that each child will have a 50% chance of having that allele. It is dominant. It will always be expressed. So, if you look at a pedigree and you see that pattern, that testing could be indicated for this family. Lynch syndrome. Lynch syndrome causes colorectal cancer, typically, although it can cause some other cancers. They're all listed below. Typically, you see it at younger ages. So most people who are diagnosed with colon cancer are diagnosed over the age of 50. And that's why the screening recommendation by the U.S. Preventative Task Force, USPTF, mostly screens people with colonoscopies at the age of 50. I know everybody hates it, but it's important for your family members to get that screening. If you're seeing it under the age of 50, in 30s and 40s, sometimes even 20s, you may be looking at a familial syndrome such as Lynch syndrome. It's also associated with endometrial, ovarian, prostate, pancreatic, urinary tract, bile duct, brain cancers. There's a few others. And it's linked to, again, DNA mismatch repair genes. Those genes that scan the other genes, like the Hall monitor gene. It is also inherited in an autosomal dominant pattern. So waiting until the age of 50 to be screened for colon cancer is not helpful to somebody who has Lynch syndrome. That's why it's important to collect family history information, even in your 20-year-olds who are there for a sports physical. Even in in an emergency room visit, you're not necessarily looking for stuff like that, and you're very problem focused. But at some point, if that patient is admitted, it's important to collect family history. And also to educate people about the importance of knowing their family history. Fanconi anemia is another cancer syndrome associated with a lot of cancers, commonly AML, which is a type of leukemia. And you can see it in a lot of young people. It's linked to faulty DNA repair genes. These would be the genes that fix the mutations that turn on the suppressor genes or turn off the oncogenes. 98% of people who have Fanconi anemia will have some hematologic abnormality that's going to progress into cancer at some point by the age of 40. About 52% will get their cancer diagnosis by that age. And the longer they live, the more likely they are to have cancer. It's inherited in an autosomal recessive pattern. So those are just some examples of cancer syndromes. How do we perform cascade testing? What is that? Well, that's when we test for a defective gene based on family history. So you start with the proband as the person, patient zero. They have a cancer. They have some kind of hereditary colon cancer. And we've identified that that's what it is. We're going to start testing with the close people, first degree relatives, such as siblings, children, and parents, to find out whether they have that gene variant. Based on the results that those people get, we may be testing people further out from that immediate family. If we test the parent of one of these people and we find that allele, maybe we'll test her siblings, and maybe her siblings' children will get tested. And based on their results, maybe their other relations will be tested. until we have identified that the gene doesn't exist for that branch of the family. Some family members might decline testing, and we may have to go further out to help people prevent or mitigate their risk. It requires that the family member discloses their diagnosis and their testing results to the family, and that's always tricky. Um, Family dynamics are hard, so you don't always know what their relationship is. Maybe this person hasn't talked to their brother in 20 years, and now they have colon cancer. Well, the right thing to do is to inform that person so that they too can be tested for colon cancer. And their children can be tested for the gene variant. And maybe, other people in that family can be tested for the gene variant. The whole privacy versus duty to warn thing started with a cancer case where a physician was held accountable for the proban's decision not to inform family. And the courts found that this physician had an obligation to family members. But that gets very, very difficult. And it's hard to know where it all begins and ends, because really, I mean, what if you find out through 23andMe you have a half sibling somewhere? There was something going on in your family. Are you obligated to tell that? Is the provider obligated to tell that person? I mean, the right thing to do is, if you know that somebody's at risk, you warn them. But how much information should we be gathering from this person? And how do we contact these people? And is it our job to violate HIPAA? And it opens up a can of worms about patient privacy. So that debate does come into play with cancer syndromes. Nurses and genetic counselors can help people find ways to disclose their diagnosis to their family members, and to increase awareness of their testing options and treatment options. Sometimes sharing the lab report with family members, giving them a copy of the lab report to share with their own providers, is important. Now we're going to talk about pedigree analysis, and that's like sort of the end of our lecture. So here is a cancer. We have breast cancer. This diagram actually came from your book, and I'll tell you the truth. If I were grading this, I probably would have to mark it down a few points, because while some of these people are marked off as deceased, if the proband is 72, sorry, let me go back to that, the chances that a grandmother who was 81 at the time of her diagnosis is still alive are bad. So there's some things missing here. But here, this proband is 72 years old. She is female. She is diagnosed with breast cancer. Going up her descendant line to her mother had breast cancer as well at the age of 68. And maybe she's thinking, aha, that means this is genetic. But if you look, her mother had 4 siblings and none of them had breast cancer. And her mother didn't have breast cancer. There was one maternal aunt who at the age of 81 was diagnosed with breast cancer. They were diagnosed at later ages. It's not a prevalence that would be more than what's in the general population. She has a paternal aunt also on a different side of the family who had breast cancer at the age of 75. This is likely to be a sporadic cancer, a cancer that arose due to aging and hormone exposure over a lifetime. Probably not familial. On the other hand, we have this pedigree, and I do have some issues with this pedigree also. There's no arrow to indicate the proband. It was like the only thing I marked most pedigrees off for. This patient, proband, was diagnosed with ovarian cancer at the age of 31. That's a pretty serious diagnosis, and typically in the general population, not diagnosed until women are in their 70s or 80s. So she's very young. She has this horrible diagnosis. And when we go up her descendant line, her mother had breast cancer, diagnosed at the age of 44, had a unilateral mastectomy. Then at the age of 54 was diagnosed again with a contralateral breast tumor and treated again at the age of 54. In both cases, she was younger at diagnosis. She has bilateral breast cancer. She's a first-degree relative, and she was younger at the age of initial diagnosis. Her 2 siblings, her sister, who was 35 at the time of diagnosis, also had breast cancer. Her brother, who typically wouldn't be at risk for breast cancer at all, was diagnosed at the age of 40 with breast cancer. So the evidence is pretty strong here. Now, if we go up again one generation to generation one, her grandmother died at the age of 36 from ovarian cancer. Again, not typical for the average population. Her grandmother's sister died at the age of 34 from some type of female cancer, most likely ovarian cancer, associated with this variation. So, and then she had a brother who died at the age of 40 from bone cancer. Although, depending on what year this pedigree was drawn, the information may not have been scientifically accurate, or maybe the report from the family member, maybe the provider knew that this was a breast cancer that metastasized to bone. So it's strongly suggestive that this is a familial cancer syndrome, and it looks like it's inherited in an autosomal dominant fashion. And this cancer happens to be BRCA1 induced. Now we have Lynch syndrome. So our proband here had a diagnosis of colon cancer at the age of 37. Looking at their pedigree, we can see their mother's sister, so their maternal aunt and their maternal uncle were diagnosed at relatively young ages from endometrial and colon cancer, respectively. The maternal aunt died from endometrial cancer, again, usually associated with older folks. And then the grandparent, the grandfather here, died of colon cancer. His diagnosis happened at 59, which is not overly young. We do not know the cause of death for the maternal grandmother. But you kind of look and it's like this person had the disease, and roughly 4 children, 2 of them had a diagnosis of a cancer associated with Lynch syndrome, so that's half. And this child had it in this generation. And the proband is 37, which is much younger. We would not normally screen for colon cancer in this person. So this person has Lynch syndrome. And that is a familial cancer, and that's what that pedigree looks like. Now, here's a pedigree analysis in cancer. Some cancers have low degrees of penetrance, so even though the gene is present, it's not necessarily leading to cancer, or it doesn't progress as rapidly. Retinoblastoma happens to be a degree with very high penetrance, not 100%, but close to it. So here we have generation 1. We have an individual with retinoblastoma, okay? They have the disorder. They marry someone without that genotype, and this couple has 4 children, 2 of whom have retinoblastoma, a boy and a girl. So it doesn't look X-linked, especially not if a female is expressing the phenotype. One of those the female child that's affected by retinoblastoma has 4 children herself, and here are the offspring. 2 of them have retinoblastoma, 2 of them do not. And in the branch of the family where the person does not have retinoblastoma, none of the descendants in generation 3 have it. So you look and you see that it's an autosomal dominant disorder. Present in every generation, can be inherited from a parent of either gender, or it can affect people of both genders. Autosomal dominant analysis. And that is what I have for you. It was a lot. I thank you. The next video series will be on genetics and aging. I don't think that will be as much information, but that kind of concludes our lecture on cancer. Thank you.